You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech Podcast. I have Mark Fischer-Colbrie. Yeah. He's with Labcyte. The web is labcyte.com, and site is CYTE. Mark, how are you doing?
Good, doing great. Thank you.
So tell me about Labcyte. What's the premise of the company?
Labcyte has invented an incredible technology that uses sound to move liquids with extreme precision and accuracy in increments to the billionth of a liter. And because the process of being able to transfer liquids in this novel way is all governed by sound waves, it is able to replicate every single transfer the same way, such that there's such uniformity and consistency to be able to generate much better scientific data and get much better results in a broad range of scientific experiments, ranging from drug discovery to diagnostics.
You said you're trying to deal with quantities of a billionth of a liter. Is that right?
Yeah, it's a situation where it has that ability to go that type of resolution, if you will. And if you think about science is all about continuing to advance precision. And this is really the first time that scientists in the life science community can really control liquids with this type of accuracy. And that's a situation where the traditional methods of moving liquids around are usually through pipettes.
And I was going to ask you, what's the lower limit on a pipette? How much can you manipulate with a pipette?
Yeah, that's a great question. People feel comfortable maybe going down to not the nanoliter level of a billionth of a liter, but they're comfortable going to the low digits of microliters. And that's a lot more liquid. And if you're trying to run experiments with expensive chemistries or with rare samples, traditional techniques just won't work. And because you're in a situation where there are many other sources of error from pipettes, where liquids are in touch with a solid surface and there are variable drops, and there's cross-contamination because pipettes have to touch a liquid in order to transfer it. Things adhere to the inside of a pipette tip, like spaghetti sauce on a Tupperware container. And you would think that you're testing something, but because you're seeing the liquids coming out, but the thing you're testing is actually adhering to the side of a pipette. All those cause so many sources of error, where with acoustic dispensing, we're able to completely eliminate that and really provide scientists with an industrialized process that completely revamps their results in dramatic fashion, such that new drugs are getting discovered that would otherwise have been missed.
Yeah, that's super interesting. What happens to the property of a fluid as you have smaller and smaller amounts? Is there you cross a threshold into some other regime or realm of behavior?
That's a great question. The bottom line is everything that's in contact with a solid surface has chaotic flow. And as you go lower and lower in volume, that chaos increases, such that at the end of the day, you don't really know what you're dispensing, because everything that you're going out through that tip of the pipette has such a variable amount of volume that people cannot control it down to that level of billionth of a liter. And so this then means that people are now able to do experiments, for example, where they can take cells from a patient who has leukemia, interrogate those cells with drug therapies that are on the market, and look for what drug or combination of drugs will be effective at killing the cancer for that particular patient. And there are leukemia patients alive today directly as a result of the precision and accuracy in the low volume dispense that has enabled doctors to prescribe the right drug or combinations of drugs to cure the cancer in those patients. So that's a new workflow that they cannot do with a standard pipette process, because you need more volume of liquid and you're going to introduce so many sources of errors, you can't get a good result from that analysis.
Yeah, when do you cross into the realm of what's called microfluidics, and what is microfluidics? How does it relate to what you do?
Yeah, if you think about it, it really is in the microfluidic regime in the context that you can think of microfluidics as a hardwired architecture, if you think of cables and channels and things like that. And you can think of replicating almost all the things you can do in a microfluidic channel as wireless with an acoustic dispensing capability, because you've got complete freedom to transfer any fluid anywhere, anytime from one location to another location with acoustic dispensing that is done in a way so quickly that you can do all kinds of experiments and generate a wide variety of results and outcomes, ranging from drug discovery work to synthetic biology to new diagnostic capabilities. And so it offers a much broader flexibility, and you're not chained to having run things through a microfluidic device that's designed for one or two purposes. We offer basically a tool that scientists can run from an experiment, say, of doing siRNA screen or qPCR or next-gen sequencing. At the drop of a hat, they can shift from one experiment to the next. And so you're not confined to an expensive microfluidic device in order to achieve your goals.
So how do you move liquids acoustically? Like what are the steps and what happens?
Yeah, it's really cool. It's magic, because you put a transducer that generates sound energy below a container that's acoustically friendly. It's got a flat bottom and certain characteristics. In this case, it's often a plastic microtiter plate. And that sound energy is then generated to create a signal where, from the reflections back, as the sound goes through the microtiter plate and through the liquid and it gets reflected back, you can basically read that data, and you know how much liquid's in the well, you know where the top layer is, you get information about surface tension and viscosity, and then we're able to dial in just a little extra power that then creates a mound, and then a little extra power has that mound fly off in a completely precise way, like a rifle shot, to a container that's inverted above it. And you would naturally think, well, if it's inverted and above it, why doesn't the liquid drop back down again? But the reality is that surface tension is much stronger than the force of gravity. So you're able to affect quite an array of transfers. And you don't have to worry at all about the liquid coming back down again. So you can shoot into a wide range of targets, of microscope slides or microtiter plates or tubes or other materials that you want to collect onto. And then you've got that and you use it for your experiments. So it's a very novel approach and use, and it's been able to generate a whole series of new workflows as a result.
So it forms sort of like a droplet that's like suspended.
Yeah, that's correct. You shoot a drop up, and it hits, say, the bottom of a microtiter plate well, and it sticks there. It stays suspended there. So you can fill up most of all the small microtiter plate wells, for example, and you don't have to worry about the liquid coming back down again. And that has huge advantages because, often people are dealing with expensive chemistries, and you can shrink down the volume, and you can save often 99% of the cost of an expensive reagent to run experiments. Or if you've got a very rare sample that you want to test, and you don't want to use microliters of the sample, but you want to use a few billionths of a liter, then that's a condition where there are tremendous advantages for using our systems, not to mention you get rid of all the plastic waste of all the millions of pipettes that are used. So as a consequence, our devices are all around the world and at the hands of hundreds of customers. And it started off in the world of what's known as high throughput screening for pharma, where they're evaluating millions of compounds a month to find out, well, what might make a good therapy. So basically all the top 20 pharma have adopted acoustic dispensing, starting off with that initial application. And now it's, everywhere in the world of genomics and proteomics and interfacing to mass spectrometry systems. And this thing is turning out to be a treasure trove of ability of providing science with great data that in turn, they'll be able to feed into artificial intelligence with much better results and better outcomes because of the tremendous precision of the data.
So, can you talk about some of the specific applications and why this works and why pipetting doesn't? Maybe drug discovery. Like, what's the difference and why?
Sure. So let's take a couple applications. One is when people are working in the world of synthetic biology, what they want to do is a lot of experiments to replicate a gene that they want to place into bacteria and have that bacteria actually make something. In this context, instead of, say, making beer, which happens through a fermentation process, bacteria can be used to make different things. And so the process of stitching together a gene can be done very, very rapidly with acoustic dispensing, typically taking the time down from 18 hours with a pipette down to 3. And then after you put the gene together, you want to validate the sequence of that gene. And then in that context, you can now, at 1/100th of the cost, because you shrunk down the expensive chemistries associated with this, run it through a sequencer and do it at a much lower cost. And so those are applications in synthetic biology. In the world of genomics, where you're doing diagnostic work, for example, and you want to do a PCR process, you can get rid of all the costs of the pipettes. You can get rid of the expensive chemistries and reagents involved in a PCR process, where you're looking for a particular gene profile as your diagnostic marker of, say, a cardiac condition. And so with that reduction in volume and increase in speed, it's just a huge savings for folks. And the applications literally run in the hundreds, and that it is a broad scale set of applications that run through up and down the world of drug discovery, from high throughput screening to toxicology work, to animal testing work, where now you can reduce the use of animals in testing, because you can now extract a much smaller amount of volume from the animal for testing. So to academic accounts who are using our systems for research, to diagnostic companies, so folks that are doing the equivalent of 23andMe, for example. That's not one of our customers, but people who do that kind of work use ecosystems from Labcyte. So there's literally hundreds of applications that this has been employed for.
So again, when the fluid gets to be that small of an amount, does it act differently than normal fluid? Does it tend to act more like a solid, or does it change its properties?
That's a great question. One of the real advantages of doing things in small volumes like that is that there's a lot of different diffusion characteristics. In the context, it's almost instantaneous. So in addition to getting highly repeatable volume, where every drop is the same as the next drop, you're in an environment where you don't have to worry about kind of mixing things together to get a good result. So that's a situation where that's an instantaneous process. And so really what ends up happening is people are able to completely reinvent workflows and do things differently than what they did before, because they're not chained to a pipette. They're not having to take a pipette off the device, either wash it or replace it, again, because pipettes always are in contact with liquids that would then cross-contaminate if you touch it again to another liquid. And so you're in an environment where people can reimagine their workflows. And fundamentally, across all these different applications, there are literally hundreds of publications out there about acoustic dispensing. And in the context that's so critical, there's a stark differentiation between data generated from acoustics versus data generated from pipettes because of this precision and accuracy capability.
Okay, very good. So what's your job? Is it just to provide the mechanism of how to do it, or the machine and the transducer that does it, or to provide analysis in your lab doing it?
Yeah, great. Another great question. So we sell the instrumentation to all of those broad customer base. We also sell the wraparound automation platform because a lot of people will require some automation for their high volume workflows. And, our machines are used both in low and high volume, but they're able to affect for pharma 700,000 transfers per day to run their experiments.
Oh, wow.
And so we also provide them with acoustically friendly microtiter plate to be able to make the transfers work efficiently, and along with service contracts. And really what we've done a lot is we started off earlier on in the company's history, we could only transfer one liquid, and that's the liquid that compounds are dissolved into, which allowed us to get into all 20 of the top 20 pharma for high throughput screening. And they in turn mandated their contract research organizations to go all acoustics because of the vast improvements in data and the lower cost. And then we figured out in 2012 how to transfer automatically basically any fluid anywhere, anytime, with understanding how to analyze the signals coming back from the sonar pulses to think about surface tension, viscosity, and other characteristics. And so basically now it can pretty much transfer anything at will. And if you think about it with a pipette, there are things that will come up a pipette, or there are things that run out of a pipette, or it won't be retained inside. And all those issues get completely dealt with automatic acoustic dispensing with a technique that we call dynamic fluid analysis, where we use signal processing to understand the liquid and automatically transfer it without any operator intervention at all.
And is there, I guess there's less, do fluids tend to interact with their pipettes no matter what material it's made of, or are there certain materials that are completely inert and don't affect the fluids?
Yeah, that's a great question too. And so a lot of people spend a lot of money trying to do that. But at the end of the day, there's about 30 different sources of error with a pipette. And no matter how much money you try to replicate that, the laws of physics will just tell you you're going to end up with a problem on just even volume delivery. So I really liken it to no matter how good you design a propeller aircraft, it can never go supersonic. So that's a direct analogy to the limitations that occur. And the entire world is trying to miniaturize, and the world's trying to get industrialized. And standard pipette technology is just not going to get you there, no matter how good you make it. And people tried all kinds of different techniques to accomplish those goals, but the laws of physics just won't let them get there.
What happens to the body of a fluid when you apply acoustics to it? Can you tunnel through liquids acoustically, or create cavities selectively, or do other things?
Yeah, we're in a situation where we're introducing such a low level of energy that for us to make the droplet, the energy level is way less than what you would do for medical grade ultrasound. Other people, for example, who use sound energy to shear DNA, you're talking much higher levels of energies imparted into a liquid. And beyond that, there are some folks who will do sound energy cause bubbles that in turn will have cavitation that, then can get super hot. But you're talking massive energy levels compared to what we're talking about for acoustic dispensing. So we can very safely transfer cells, DNA, RNA, with no impact. In fact, it's more gentle to transfer a cell with acoustic dispensing than what happens when you go with shear stress through a pipette tip. So it's actually a gentler process than that.
Okay, and then would with this acoustic movement of liquid, would it be useful to mix tiny amounts of two different substances, evenly and efficiently?
Yeah, the applications just are really amazing. And part of what we do, separate from providing the instrumentation, is we work closely with people to develop these applications and new workflows for folks. And so, for example, we're not just precise and accurate in terms of volume, but we can also basically aim the drop. And so if you want, you can shoot onto a microscope slide, again, upside down, and you can spot it with, say, cell media, 1536 drops, in an array format. Then you can come back and you can transfer cells into that cell media, and then you can come back and you can transfer a compound into that, so you can run, 1536 mini experiments in an array structure on a microscope slide. So this is the type of thing where we have a large applications team that's out in the field that works with customers on creating new workflows. Another example is we've been able to do direct injection into mass spectrometry systems with a few nanoliters of materials. And that's not yet a commercialized product, but AstraZeneca have run already 12 million tests using this approach, and have had fabulous results, which is another opportunity to have a complete game changer in the world of drug discovery and diagnostics. So really interesting things can happen when you miniaturize, and really the only effective way to miniaturize is with acoustic dispensing.
Okay. Well, very good. So what's the best way for people to find out more about what you do and to get in contact?
Yeah, if you go to labcyte.com, there's a really great website there in terms of looking at the breadth of what we do. There's a lot of great videos, a lot of wealth of information around technical notes and application notes. We've got references to hundreds of publications. So there's really a tremendous amount of data out there, as this has been well adopted really since 2004, and one where we're just excited that there are tremendous improvements that have gone on with precision medicine, and with this opportunity to treat cancer patients, with new drugs getting discovered and new diagnostic tools being created that are having a real impact, and we're just thrilled to see that.
That's great. All right. Well, very good. I appreciate you for coming, and people should go to labcyte.com, CYTE.
Yeah, labcyte.com, and check out the great videos there.
That's great, Mark. Well, thank you again for coming.
Thank you. Appreciate your time.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.